Christian Frisch is R&D Manager at ABD Serotec and has been head of the Antibody Generation Group since 2003. He studied molecular biology in Göttingen, Germany, and did his PhD with Professor Alan Furst at the University of Cambridge in the UK, working on the energetics of protein-protein interactions and protein folding. In 1997, he joined MorphoSys, working on the development of display technologies and the generation of antibody phage display libraries. His group at ABD Serotec has generated more than 20,000 antibodies in over 1,000 custom projects over the last 10 years. Over to you, Christian.
Yeah, thanks, Amanda. Okay, I would like to show you today how you can improve your antibody drug development assays with anti-idiotypic antibodies, and I will explain how we at Bio-Rad ABD Serotec generate recombinant anti-idiotypic antibodies. I will start my presentation with a definition of anti-idiotypic antibodies, and then I'll describe the two major assays in which these antibodies are used: immune response or anti-drug antibody assays and pharmacokinetic assays. I will then introduce you to the three different types of anti-drug antibodies and how these are generated by guided selection using HuCAL phage display technology. In the last part of my presentation, several examples for the use of HuCAL antibodies in IR and PK assays will be shown. So anti-idiotypic antibodies are antibodies that are directed against the idiotope, which is the unique set of antigenic determinants of the variable portion of an antibody. The idiotope consists of the antigen binding site, the paratope, as well as regions near the paratope. The paratope is the region that binds to the epitope of the antigen. So anti-idiotypic antibodies are specific for one particular antibody, for example, the drug antibody. And they are inhibitory if they bind the paratope, since then they compete with antigen binding. So one of the major assay types where anti-idiotypic antibodies are used are immune response assays for detection of anti-drug antibodies in serum. Protein drugs are frequently immunogenic and lead to development of anti-drug antibodies. And even humanized and human antibody drugs may elicit a humoral immune response, and these anti-drug antibodies may be neutralizing the drug efficacy or create safety issues. Therefore, for preclinical and clinical development of antibody drugs, innovator drugs as well as biosimilars, the sponsor must show the immune response to the drug. And these anti-drug antibody assays require reference standards, which are ideally human anti-drug antibodies, which cover a broad range of affinities and bind to varying epitopes. In some cases, also different isotypes or antibody subclasses are needed. And what is, of course, also very important is a consistent supply over many years. The second major assay type where anti-idiotypic antibodies are used are pharmacokinetic assays for quantification of human antibody drugs in serum. And this is a little bit like finding the needle in the haystack, because in human serum there's 10 milligram per milliliter IgG1, and your drug can be in the concentration in the nanogram per milliliter range. So you really need to detect your drug specifically within a million-fold excess of very similar molecules. And to do this, you can either use the antigen, so the drug target, or anti-idiotypic antibodies. And on the lower side are typical assay formats, which include just capturing the drug out of human serum using the antigen, and then detection with an labeled anti-FC antibody. But in these assays, you often face background issues. So it is better to use two specific molecules, both as capture and detection, as in the other two examples, where either two anti-idiotypic antibodies are used or an anti-idiotypic antibodies in combination with labeled antigen. I would now like to show you the different binding modes and properties of anti-idiotypic antibodies. We've classified anti-idiotypic antibodies into 3 types, 1, 2, and 3. And the type 1 is a paratope-specific anti-idiotypic antibody, which is inhibitory and neutralizing. But this antibody only detects the free drug. So if there's a large amount of drug antibody in the serum which is still bound to drug target, then it is more useful to use the type 3, the type 2 antibodies, which is actually non-paratope specific, so also not inhibitory. So this antibody, anti-idiotypic antibodies, can still bind the drug even when the drug target is bound. So this type detects total drug, free, partially bound, and fully bound. And then we recently came up with a third type of anti-drug antibody, and this one is drug target complex specific. We call it type 3. It is not inhibitory, and it exclusively detects the bound drug. And I will show you in the next slides examples for all 3 types and how we distinguish these in different assays. The first assay we use is typically an ELISA, and here is an example where for Golimumab we found all 3 types of anti-idiotypic or anti-drug antibodies. And as you can see, the type 1 antibody is very specific for Golimumab. It doesn't bind any of the isotype controls, which are also IgG1 kappa antibodies. And it does also not bind to the human TNF-alpha Golimumab complex. On the other hand, the type 2 anti-idiotypic antibody binds both to the free Golimumab and also to the Golimumab human TNF-alpha complex. While the type 3, the complex-specific antibody, does not bind to any of the free antibodies, but only to Golimumab when complexed with human TNF-alpha. Another assay to discriminate the different types is an inhibition assay. And here is an example for 2 anti-idiotypic infliximab antibodies. And they were tested in an inhibition assay. A constant amount of infliximab was incubated with different amounts of anti-idiotypic antibodies. And after incubation, free infliximab was quantified on immobilized TNF-alpha. And what is very apparent, I mean, the type 1 antibody, the ABD17841, this can inhibit the binding of infliximab to TNF-alpha. While the type 2 antibody, the ABD19370, shown in red, does not influence the binding of the drug infliximab to TNF-alpha. So this one clearly must bind to an epitope outside the paratope region. Another assay to show that these different anti-idiotypic antibodies bind to different epitopes was done by real-time kinetics, an epitope binning experiment, where infliximab was coated on a sensor and then incubated with the type 1 antibody ABD17830. There is a nice binding, and after saturation, the sensor was dipped into solution containing a second anti-idiotypic antibody, the type 2 antibody ABD19370. And you can clearly see a nice additional binding, and this is the same as if this antibody was applied as a first antibody in the control experiment. So these 2 antibodies clearly bind different epitopes. Now, how do we generate these different anti-idiotypic antibodies? We use a recombinant technology, phage display library, which is called HuCAL Platinum, and HuCAL stands for Human Combinatorial Antibody Library. This is a synthetic library. It uses human antibody genes as building blocks, and these antibody genes cover the structural and sequence diversity of the human immune repertoire. We used a modular gene design for library generation. All the 6 CDRs are diversified with high quality trinucleotide CDR cassettes. The library is very big. It contains 45 billion functional human antibodies, and we use a proprietary phage display approach, namely cis-display, which is ideal for selection of high affinity antibodies. Since HuCAL is modular, affinity maturation can easily be performed by simply exchanging one CDR with a CDR cassette. Also, the selection from HuCAL is very straightforward and fast. You simply mix your phage library with the antigen and then perform 3 rounds of phage display, and here you can guide the selection into the direction you want, and I will show you examples for this in a few slides. After the 3 rounds of phage display, the whole pool of Fab genes is subcloned into an expression vector, and then single colonies, so monoclonal antibodies, are screened. We usually do that in a 384-well format. The best hits are then sequenced, and only unique antibodies are expressed and purified and tested again. So after 8 weeks, you have your Fab antibodies, which can be either monovalent Fab or bivalent Fab, and you also have a choice of different tags, which are at the C-terminus of the FD chain. If you want, you can also convert these Fabs into different IG formats later on. Now, the generation of the anti-drug antibodies. I mentioned already the guided selection, and this is very useful actually for selection of the different anti-idiotypic antibody types. For type 1, we immobilize the drug antibody on magnetic beads, for example, and then incubate this with the phage antibody library, and at the same time we add isotype matched control antibodies in human serum to block away all the antibodies that bind to the constant regions of the drug. So the focus is then really on antibodies which bind to the binding site of your drug antibody. For type 2 and type 3 anti-idiotypic antibodies, the selection is a little bit more complex. Here we do the selection on the drug target complex. So for example, we would immobilize the drug target and then incubate with the drug to form the complex, and then apply the HuCAL phage display library together with isotype matched control antibodies. And here, in addition, we add the drug target because we also don't want to generate antibodies against the drug target. I would now like to show you examples of antibody generations against drug antibodies, and also in this slide explain how we generate antibodies, or how we select for antibodies with the best affinities. So we've recently included in our selection process an off-rate screening, and this is an example for an off-rate screening of selection against Cetuximab. We took 95 hits from the primary ELISA screening, which were specific for Cetuximab, and tested these or measured the off rates of all of these 95 clones, which can be very easily done using the crude Fab-containing E. coli lysates, the same lysates that have also been already used in the primary ELISA screening. And what you can see here that we have quite a diversity in off rates, which could actually be confirmed by measuring the affinities of the purified Fabs. So what we did is we sequenced all the 95 clones and ended up with 56 different antibodies. And sequencing of the 20 best clones from this K-off ranking that I've just shown would have resulted in the 7 antibodies shown in red. So clearly, from this K-off screening, the antibodies with the best off rate have been selected. What is also apparent is that we have a high diversity of antibodies, and the antibodies cover a broad affinity range from 500 picomolar to 174 nanomolar. Now, this doesn't need to be the end. As I mentioned in the beginning when I introduced you to the library, it's a modular design. So if the affinity is still not high enough, it's quite simple to improve the affinity by performing an affinity maturation. And to do that, we exchange one of the CDRs, usually the LCDR3 or the HCDR2, with a highly diverse cassette that we have in the freezer pre-made. So generate a new library and then do another panning with higher stringency. So, for example, a higher washing stringency, many washes, longer washes, or also a reduction of antigen in the different panning rounds. And then we usually end up with affinity matured unique antibodies. And to show you an example, we've done that, for example, with one of the with anti-idiotypic antibodies. Here, different anti-idiotypic antibodies from a standard panning against Trastuzumab are shown. All these 11 antibodies are highly specific for Trastuzumab, and they already have quite good affinities in the low nanomolar to even picomolar range. But we still wanted to improve the affinity of the antibodies which performed best in our assays, and that were the 16712 and the 16714. So we did exactly the procedure that I've just described, exchanging CDRs and then performing the affinity maturation panning, and ended up with several candidates, 56 new hits which we analyzed, 26 for 16712 and 30 for 16714. All matured antibodies are still highly specific for Trastuzumab, as shown in the ELISA, and the affinities of the derivatives are now down to 70 picomolar for the ABD 16712 derivatives, and even down to 20 picomolar for the ABD 16714 derivatives. So maximum affinity improvement in this maturation was 120-fold. And what is also apparent from these data here, the parental antibody, which had already a better starting affinity, there are some derivatives which actually have a lower affinity now. So for these antibody, it would have been probably better to use even more stringent conditions in the panning. But these were both performed in the same maturation package using the same conditions. Now I would like to show you PK and IR assay data. And the first assay data is actually with the antibodies from this maturation, and showing you that these high affinity antibodies also lead to very sensitive assays. It's a PK assay in the bridging format. We use one anti-idiotypic antibody as capture antibodies and in the Fab format, and as another anti-idiotypic antibody in the IgG format, HRP labeled for detection. The capture antibody is 70 picomolar and the detection antibody 20 picomolar, and this translates into a very good assay sensitivity down to the about 100-200 picogram per milliliter range, Trastuzumab in 10% human serum. Now an assay, same assay setup, PK assay in the bridging format, but here's an example where we mixed two different anti-idiotypic types, namely a type 2, which was used for capture, and the type 1 used for detection. And if we now use these two types, which bind to different epitopes on the drug, we can use a higher coating concentration of the capture Fab, and this way it's possible to also in this assay reach a very high sensitivity also in the 100-200 picogram per milliliter range in human serum, although the antibodies don't have such a high affinity as in the assay that I've just shown. Now the next assay I would like to show is one with a type 3 anti-drug antibody. In this case, an anti-Adalimumab anti-drug antibody. And first of all, I would like to show data showing that this ABD 18754 is highly specific for Adalimumab complex with TNF-alpha. It does not bind to TNF-alpha and Infliximab complex, to TNF-alpha alone, or to any of the other drug antibodies alone. And with this type 3 antibodies, you can do special PK assays, which is not possible with the other anti-idiotypic antibodies. So here you can use, you can coat your drug target, in this case TNF-alpha, at higher concentration, and then capture the drug out of the human serum and detect with a labeled type 3 antibody, and also here reach very good assay sensitivities. Now to the, if you want to use these anti-idiotypic antibodies in immune response assays as reference standards, they have to be converted to full-length human immunoglobulin. And this can also be done very easy with recombinant technology. We know the sequence of all of our antibodies, and it's a simple cloning exercise to transfer the Fab antibody genes into the respective vectors for expression of human IgG1, 2, 3, or 4, or human IgA1, IgE, or IgM, and then express the antibodies in mammalian cell culture. I have two examples for the use of these antibodies in immune response assays. In this bridging format where the drug is coated and detection of the anti-idiotypic antibody or if the anti-drug antibody is with labeled drug. So the anti-idiotypic antibodies in this assay are titrated in 10% human serum, and we used here 3 anti-idiotypic antibodies with different affinities from 160 picomolar to 10 nanomolar. And what's very apparent is that the affinity clearly correlates with assay sensitivity. So here you would you can sort of mimic the natural immune response by using these different anti-idiotypic antibodies as reference standards. Another example, same assay set up for Cetuximab, where we've also generated antibodies in an affinity range between 500 picomolar and 10 nanomolar. And also here, clearly the affinity correlates with the assay sensitivity, with the antibody with the best affinity having the highest sensitivity in this immune response assay. So let me summarize the custom service for anti-idiotypic antibody generation. First of all, it's a very fast process. In only 8 weeks, we can deliver the antibody in the Fab format. We can drive the selection towards the antigen binding site of the antibody by using framework-matched controls. We can select drug-antibody target complex binders or non-paratope-specific anti-idiotypic antibodies. We do not have any matrix effects of human serum through addition of human serum during the selection process. As I've shown you, we can achieve very high affinities, leading to sensitive assays. And the affinities of the anti-idiotypic antibodies cover a broad range, which can be useful as reference standards for immune response assays, where the clinical responses vary and may sometimes be of modest affinity. And the anti-idiotypic antibodies can be converted into fully human IgGs, IgA, IgM, and IgE. We do not only offer this as a custom service, but we also have ready-made antibodies to biotherapeutics in our catalog. And here's the most recent list of anti-idiotypic antibodies we offer. We have for the different drugs both products available in Fab format and IgG format. For some, as I've shown the example for Adalimumab, we have all the 3 different types, type 1, type 2, and type 3 available. The anti-idiotypic antibodies in most cases cover a broad range of affinities. And we have supporting data for all of them. We have PK and IR assay data, and for most of them we also have inhibition data. Now, a few questions came in during the presentation, and I will now review these questions and hand over to Amanda.
After this webinar, if you have any other questions more appropriate for Christian directly or for me, please feel free to contact us directly using these details.
So there was one question regarding planning the launch of additional anti-idiotypic antibodies, or whether we add the blank spaces and add type 2 and type 3 antibodies against drugs we already have in the catalog. The answer is yes, we are working on new projects, for example, Panitumumab, and we'll launch them in the near future. There's also ongoing projects to generate type 2 and type 3 anti-drug antibodies for some of these where we haven't got them yet. So we will constantly add more products to this list. Then there was another question to type 2 and type 3 antibodies. How difficult is it to generate these? It's certainly more challenging than generating the type 1 antibodies, as there are fewer epitopes available. But also here in most cases we are successful, but there are simply fewer antibodies selected. Type 1 basically always works, and we have a large variety, as you've seen for the Cetuximab example, to choose from. And then maybe as a last question, how often do you need to do an affinity maturation to achieve the required assay sensitivity? For the anti-idiotypic antibodies, so far it was not really necessary to perform affinity maturation. The required sensitivity of the assays is in the low nanogram per milliliter range, and this has been achieved routinely with antibodies from our standard process. So the maturation I presented here was more academic interest, but I think it's good to know that it's possible to improve the affinity of a HuCAL antibody. I think these were the most pressing questions. So that's it for today, actually. Thank you for your questions and for attending the presentation. Goodbye from all of us.
Thank you. That does conclude our conference today.